Okay, we'll start microbiology chapter 10 today. And let's see, we'll try to get through 10, 11, maybe 12 today. Dr. Sloan will be here next week and the week after for the micro class. I'll also post those lectures up on a media format for you as well. I'll post them up ahead of time so you'll have access to those. But I'll be out of town next week and the week after, and Dr. Sloan will be substituting for me. The first lecture he talks about will just kind of be about epidemiology and microorganisms, how do you track diseases. And then the second lecture that he does will cover diseases in the gastrointestinal tract. So it'll be a little off schedule on the syllabus. I'll make the corrections on the syllabus so you know what he's covering. And then, like I say, I'll supplement that with some media presentations for the other material that should be covered at the same time. But he'll do those two lectures, again, one on epidemiology, another one will be on digestive diseases, microorganisms that are involve the digestive system. So that'll be for next week and the week after. Now, you'll still be required to do your media portion for this class and the lab. So, again, there'll be some lectures you'll have to listen to. And then when I return back after 2 weeks, we will go strictly into the diseases that affect systems of the body. So just give you kind of a preemptive strike on what we'll be covering in the lecture to come. So we talked about recombinant technology last week. We'll talk about microorganisms and the classifications of microorganisms today. The science of classification of microorganisms is referred to as taxonomy. You need to be familiar with this definition. It basically looks at the science of classifying organisms, provides a universal name for organisms, also a reference for identifying particular organisms as well. Organisms are in a system of phylums or a systematic of phylogeneity. This basically is the study of an organ. I guess as we look at things as in microorganisms, we want to look at an evolutionary history of these organisms. Where did they derive from? What have we done historically to be able to identify particular organisms? All species have been inventoried, all species that we know of, I think is better said, inventoried between 2000, it's believed between 2000 and eventually through 2025. We hope to be able to identify all species of life on Earth. But if you remember our earlier discussions, we talked about organisms evolve, and certain microorganisms evolve as well. Whether that's possible or not, whether there's a finite number of organisms that exist in the world as we know it, is still to be seen. There are recesses on the world, undersea areas where volcanic activity exists that we don't know what life forms exist there, so whether or not that's really possible or not over the next 12 to 15 years, I guess, is yet to be seen. Obviously, there's an evolutionary pattern, though, as far as the development of the categories that organisms have been placed in. This slide just basically demonstrates, specific milestones that have occurred over the years in development. Organisms are basically arranged into specific categories referred to as taxa. This reflects the degree of relatedness. In other words, how microorganisms may be related to one another. The evolutionary history of a group is basically referred to as systematics, or the phylogeneity or the phylogenicity of a particular organism. There are new techniques in microbiology that look at or have revealed that there are two types of prokaryotic cells and one type of eukaryotic cells. If you remember again from earlier lectures, we've talked about the prokaryotic cells basically as being more primitive, more rudimentary type of cells, whereas the eukaryotic cells are more advanced. Particularly, this depends on comparing a sequence of nucleotides. The DNA sequences, the RNA sequences of these particular organisms will help us to basically put them in the particular domains in which they exist. The cell types generally are known as the domains. And the domain for bacteria include many of the non-pathogenic prokaryotes found in soil and water, as well as some of the pathogenic organisms in the photoautotrope prokaryotes. And this chapter just kind of goes into how these microorganisms are categorized. The domain archaea, we've alluded to before, includes prokaryotes. They've never have a peptidoglycan in their cell wall. If you remember, this is the characteristic that helps to determine the gram staining of various organisms. They often live in extreme environments, as do members of the other three major groups. Methogens, these are extreme halophiles, and then the hyperthermophiles, which will live in extreme temperature areas as well. The domain of eukarya basically contains kingdoms of animals, plants, fungi, protists. These are single-cell animals that exist. Your book basically goes through the characteristics of archaea, bacteria, and eukarya. Basically, you should be familiar with this chart. Gives you a nice example of microorganism and the particular characteristics, looking particularly at the cell type, then looking at the cell wall, the constituents of the cell wall, the membrane, lipid arrangement, then whether or not amino acids, and I'm not that I'm going to ask you to identify the specific amino acids, but you can see the similarities between the various types of organisms in these domains. Antibiotic sensitivity, the RNA, whether or not it's present or not, and then the common arm of the transfer RNA. Just gives you some general characteristics that help you to put each of these microorganisms in a particular characteristics. These are the three domains: bacteria, archaea, and eukaryote, and then gives you the organisms in each. You should be familiar with these organisms' characteristics in each of these three domains. And again, this is out of your textbook. The last slide, I'm sorry, I threw it too quick. Prokaryotic cells versus eukaryotic organelles. You're familiar with the organelles, but you also need to be familiar with where do you see particularly these organelles. Obviously, the eukaryotic system has mitochondria, which we're much more familiar with, chloroplast, chlorophyll containing, and as you can see, the prokaryotic cells, you don't really see those structures prominently there. Simple eukaryotic organisms mostly are unicellular, grouped in the kingdom Protistus. Also, the kingdom of fungi includes unicellular yeast, multicellular mold, mushrooms, and nutrients that are absorbed in those categories. The kingdom of plantae, or the plants, include algae, all the mosses, the ferns, the conifers, and flowering plants. All of these are multicellular organisms. Then energy from photosynthesis and carbon dioxide and water is converted into organic molecules as used by the cell. The process of photosynthesis we've talked about previously as one of the steps in metabolism for specific certain categories of microorganisms. Then Animalia basically includes sponges, worms, insects, vertebrae. And we'll talk in a lot more detail about each of these types of organisms. They basically obtain nutrients and energy by ingesting organic matter. All eukaryotes, as discussed in chapter 12, you'll see derive this. A lot of the human diseases, especially when we talk about nematodes, parasitic types of infections, will be from organisms that fall from that category. Then classifications of viruses. We're familiar with viruses because of many of the disease processes that they cause. Viruses are not classified in any of the kingdoms, and the prokaryotes are grouped. We see viral species as populations of viruses with similar characteristics, morphology, genes, enzymes occupying a particular niche in the ecological system. Then we also identify microorganisms based upon the morphology characteristics. There are hundreds of bacteria that appear to be in shapes of rods and small cocci, which we've talked about already previously. You've also had an opportunity to identify some of those on your labs. Many of these characteristics still, some are used in the characterization or the classifying of certain bacteria, and sometimes the presence of endospore or flagella, which is why we pay particular attention to microorganisms with endospore, why endospore stains are important to us, why understanding the placement of flagella is important, because we can use the presence of these types of structures in helping us to classify and or identify particular microorganisms. So I'm not making you learn that because I'm stuck on endospores, but it will help, again, in the identification of certain microorganisms. Again, I just want you to get a general appreciation for it. I don't expect you to be running these types of experiments inside a lab at a hospital to identify organisms, but you need to appreciate what's involved in being able to identify it. And then also the structures, such as the endospore, as we've seen, and even the flagella to a degree, help these organisms as far as their virulence factors, helps these organisms to be able to survive in extreme types of environmental conditions. One of the first steps in identifying is the differential stain. You're already familiar with the Gram stain and acid-fast stain. There are also other biochemical studies that look at the enzymatic activities of microorganisms. In other words, what particular types of enzymes or enzymatic processes take place inside this microorganism. What type of chemical reaction can you see or observe in a laboratory setting? And if you observe that reaction, it helps you to know that that particular microorganism exists. You can also measure the quantity of certain enzymes or byproducts of enzymes, and it'll help tell you what the quantity of microorganism in a particular medium may be. So these tests are important. They can these certain tests can determine the ability of certain organisms to ferment. Your lab experiment today went through the process of fermentation. Not today, this week, I mean. Went through the process of fermentation. As you recognize, we discussed fermentation before. An assortment of carbohydrates are particularly utilized in this type of a process. Bacteria may also be subjected to a number of other biochemical tests to be able to determine particular mechanisms of metabolism and, again, the enzymes that may be used in these metabolic sequences. The use of selected or differential medium we've already discussed as well. They've been discussed in bacterial isolation and identification. Medically important bacteria have rapid identification tests that we utilize in a clinical setting all the time. The result of these tests, certain organisms may be assigned particular numbers to demonstrate whether or not it's positive or negative for a given result. Test results can be assigned a number of different identifiers. This is using a number system, which is basically reliable. It's important in a number of tests as well. The system sometimes is referred to as a numerical identification. You can have an alphabetical identification. You can have almost any type of identifying pattern that you want to use in a laboratory setting, as long as it's consistent for whatever the laboratory procedure is. So that sometimes varies from lab to lab. Obviously, there's a standard, so universally, lab professionals from particular areas, scientists, microbiologists can work together. But if you have an individual lab that you're running a specific sequence of experiments to try to identify, say, a particular organism that you wanted to culture out specifically, you may have your own way of tagging or identifying particular groups of microorganisms. Phages or phages, however you want to pronounce that, basically are bacterial viruses like antibodies. They're highly specific to species and or strains with species. By means of stock or different phages, you actually can get characteristics that are helped to use that may be helpful in identifying particular bacteria and gives you an ability to be able to trace particular diseases, especially in the cases of epidemics. Phage typing also is commonly used in outbreaks of illness, especially those caused by Staphylococcus. Commercial systems can help identify bacteria based upon their cellular fatty acids. That's just one of many constituents that can be identified in identifying particular bacteria. There's also a technology that's used in labs called flow cytometry. This is where bacteria can be identified without culturing, so you don't have to spread it on the culture medium. You basically use flow cytometry by moving fluids containing bacteria are basically forced through a small opening. A laser beam then is scattered by the bacteria, which gives information about the bacteria based on its fluorescence, whether it reflects a certain type of light, absorbs a certain type of light stream, and allows you to be able to identify particular organisms based on this technique. Techniques also are able to suggest evolutionary relationships amongst bacteria, determination of the DNA-based sequencing. We've gone over the importance of that before. So when particular organisms have a consistent relationship between particular base pairings, there's a demonstration there of a relatedness between particular microorganisms. Restriction enzymes, these can be used to cut out bacterial DNA into certain base sequences. If you use these particular fragments, and then place it under a technique referred to as electrophoresis. The DNA pattern of different organisms can be compared. Electrophoresis allows certain bands of these nucleic acids to settle and give actually a map or a fingerprint of the DNA sequences of particular microorganisms. And this is as identifying as a fingerprint is for an individual, and thus is referred to as DNA fingerprints for microorganisms. Utilizing, again, a specific restriction enzyme, it cuts out portions of the DNA, and then those portions of the DNA have a certain weight, and utilizing electrophoresis will give a particular pattern that can be read to identify microorganisms. The other study that we talked about before, the polymerase chain reaction, it basically used to identify, or it's basically used to increase the amount of microbial DNA to the level that it can be detected. So if you have a small sample of an organism, you can use the polymerase chain reaction to give a larger sample of the DNA that's being that you're trying to identify and use. If double-stranded molecules of DNA are heated up, complementary strands will separate. We know heat, changes in pH will alter the DNA structure. The hydrogen bonds that connect the bases together will then break apart under these types of conditions. And because the strands are complementary, they'll unite if they're allowed to incubate together over time. So if you separated the strands of these different organisms, incubated them together, they have a close relationship. The greater the amount of DNA that will join together, you'll have a greater amount of complementary base pairing. This is called hybridization, or nucleic acid hybridization. It assumes that two species are similar or related somewhat, and a major portion of the nucleic acid sequence will be similar. There's another technique referred to as southern blotting. Again, we're identifying particular microorganisms. Southern blotting makes use of the nucleic acid from hybridization to identify particular microbes. DNA probes are used on this technique, being developed to help rapidly identify particular bacteria. This is used commonly in laboratory studies as well. Now, you should know the definition of dichotomous key. Information considering points of microbial identification are basically placed into a dichotomous key. The workers will answer a successive number of questions which will help to direct the individual to the next step. There are informations are kept in formats referred to as cladograms, which are basically maps that demonstrate evolutionary relationships between various microorganisms and how they evolved. Each branch point is defined by a particular feature in the bacteria, and these sequences, RNA sequences, have been used to make the cladograms and to give the information about the base sequences of various types of microorganisms. All of this is basically used to try to help identify microorganisms. Knowing the historical data about what made up these particular microorganisms will further help identify these particular types of microorganisms. So we also want to talk and recognize the scientific names of various microorganisms and how they're related. In species definitions, we talk about eukaryotic species. These are groups of organisms that are related as a breed amongst themselves. Prokaryotic species, again, a population of cells with similar characteristics. Clone refers to a population of cells that are derived from a single cell. You've probably heard about cloning in the media, about how you can copy or make an individual cell, make populations of either individual cells or individuals in this case, which are derived basically from a single cell, as long as you have the genetic information that would allow you to do that. A strain refers to genetically different cells with a clone, and then a viral species refers to a population of viruses with similar characteristics that occupy a particular niche in the ecological system. The terms in the domain of eukarya that are important to be familiar with: Animalia refers to multicellular organisms. They don't have any cell wall. Chemoheterotrophic in nature. Plantae are multicellular organisms. Cellulose for the cell wall. Usually are photoautotrophic because they utilize photosynthesis. Then fungi, which are chemoheterotrophic. These are unicellular or multicellular. organisms, cell walls made up of chitin developed from spores and hyphal fragments, and then protista, which is basically everything else, eukaryotic organisms that don't fit into any of those above kingdoms. Now, the familiar characteristic that you see in each of those particular organisms or class of organisms are the multicellular. Each one is a multicellular microorganism. And this is just an excerpt out of the table I originally showed to you before. Now, some other identification methods we've talked about previously, at least alluded to in the previous lectures. Morphology, characteristics, cocci, bacillus, clusters of cocci, chains of cocci, streptococci, all are morphology patterns that you use in the identification of particular microorganisms. Knowing this morphology helps to quickly eliminate other microorganisms. As far as if we're looking at particular human diseases, if you know the morphology, it immediately excludes certain classes of organisms just by knowing the morphology. Then the next step may be to do a gram stain. If you know the gram stain of a particular organism, you can exclude multiple organisms. Taking this sequence of steps helps you to be able to rapidly identify microorganisms. It's important in disease-causing microorganisms to be able to use these identification methods rather rapidly because it helps you in the diagnosis and eventually the treatment of particular organisms, particularly those that cause disease. Now, I recognize that I said earlier in the lecture series that only a small number of microorganisms cause disease. That's true. But our focus is now going to change a little bit from knowing that we have microorganisms in our environment to where we'll specifically look at microorganisms that cause disease starting next week. So again, the tests that we've done thus far to identify certain microorganisms, now we're going to use those tests specifically to help us to identify diseases or organisms that cause particular diseases. Again, the differential staining we've talked about, gram staining, acid-fast staining, the biochemical tests, again, to identify particular enzymes and or byproducts of particular growth. This is just a microbiology requisition. You'll see this used in a laboratory setting. Many of these are computer-generated forms now. You still may have to fill some out by hand. And basically, the lab has its responsibility to record the date particular organism was received to the lab, where the lab is located at, given hospital, the date that the lab specimen was sent, the date and time. You also want to know the collection time. All of this is important because you'll see when you work in a clinical setting, you collect a urinalysis. It's not labeled. It doesn't have a person's name on it. Or let's say you put the name on it, but when was the specimen collected? Was it collected today? Was it collected 2 days ago? And when it gets down to the lab, the growth of microorganisms versus today versus from 2 days ago would give you a much different pattern of the number of microorganisms that are there. If it's not a fresh sample, you may be aggressively treating a patient for an infection that may have been a mild infection, but if the specimen was brought down 2 or 3 days later and there's been several growth cycles in that specimen, or what if this was an anaerobic type of an organism, the specimen was collected, left in an oxygen environment, and many of the microorganisms that cause the disease have died, and so the specimen gets sent to the lab, and you get an erroneous report that says there are no organisms present in the growth, but it's because the sample was collected at a different period of time. So it's important with the lab specimens that are collected, especially specimens that are collected from patients, whether that's a blood sample that you suspect a microorganism being in the blood, whether it's viral or bacterial, whether it's a sputum specimen that contains maybe viral antigens and or viral antibodies, or a specimen that has active bacteria that's in the sputum specimen, has to be dated, has to be timed. Typically the name of the physician that ordered the study, ordered the sputum to be collected, who was it collected by, and then identification information on the patient, including the patient's name and patient's ID number. And then what type of study do you want performed? Now, many specimens collected in a hospital may just be sent down to the lab, and you feel the lab will determine what's the best study to be done. Sometimes it works like that, but most of the time in a hospital setting, if an order has been to collect a sputum, you want the sputum for culture, sensitivity, and gram stain. And typically on the lab form, you have an opportunity to identify or check what you need to, what type of study you want run. If you collect a stool specimen, you may only want to check it for Clostridium difficile. But Clostridium difficile is not it's the problem, but it's actually the toxin for Clostridium difficile that you want. So you'd have to mark that lab specimen accordingly. The source of the specimen is important as well. We make some assumptions when you receive a lab specimen, but it's always better if you know exactly, was this collected from blood? Was this collected from cerebral spinal fluid? Cerebral spinal fluid is clear in appearance. If it has an amber hue to it and you send it down to the lab, it could be confused for urine, and so you would get a completely different set of test results when you compare cerebral spinal fluid to urine. Also, peritoneal fluid may be obtained from abdominal fluid, may be obtained from lung fluid. All of it will give a similar gross appearance when you look at it as the specimen is received. When you look at it microscopically, or if you do particular cultures and it's from a different area, then it would help determine what type of studies need to be performed depending on where these specimens were collected from. So almost always you need to be able to identify the source of the specimen. And then what is the test, again, that's requested? And this just goes through a number of different types. I talked about the toxin assay for Clostridium difficile. As we look at bacteria cultures, you may want anaerobic cultures, you may want a gram stain done, whatever the particular type of organism is, and this helps to break it down into specific organisms. Acid-fast bacillus, we check if we were looking for tuberculosis in a sputum specimen, so forth.